Hello everyone, I'm Miss Davis and welcome to Biology 124 at New Mexico Junior College. So let's get started. Taxonomy is a very important branch of biology, which focuses on identifying, classifying, and naming organisms. Linnaean classification is the grouping of organisms into categories. The largest, most inclusive category is the domain. Then we can continue down to kingdom, phylum, class, order, family, genus, and finally the least inclusive, which is the species. These categories create a hierarchy, and the organisms that fill a particular category are distinguished from other organisms by a set of characters. A character is any trait. It could be structural, molecular like DNA, reproductive, or behavioral that distinguishes one group from another. Here is an example of how this classification system works. You can see that a cat and you as a human are in the same domain, Eukarya. We're also in the same kingdom, Animalia. The phylum is also the same as Chordata because these are characteristics we share with cats. Class Mammalia is also the same. But after class, you see that we have a difference that starts to come. This is where our shared characteristics end. Now, for the cat, the order is Carnivora, family Felidae, genus Felis, and finally the species is Felis catus. Us as humans are in the order Primates, Homidea, genus Homo, and species Homo sapiens. You will notice that Latin is the language used here in this classification. This is because it is a dead language, and it's not changing. Our language is constantly changing. Think about it. The English you speak is not the English that your grandparents spoke. Our language is constantly evolving with new slang words all the time. You will also notice that the species has a two-part name. This is called binomial nomenclature, and it was developed by Carolus Linnaeus. In biology, it is important to use the scientific names. This is because the common names can sometimes be confusing. It's like the term locust. Locust, are you talking about a grasshopper or are you talking about a cicada? Okay, so for some people it might be the grasshopper, as others it might be cicada, so it can be confusing. So common names are confusing. We use scientific names for biology. A lot of you probably learned the kingdom as the largest group of classification, but recently the category of domain has been added. There are 3 domains that you should be familiar with. The old bacteria kingdom, called Monera, was divided into 2 domains. Bacteria is the first domain, and it's made out of prokaryotes, cells without a nucleus or various organelles. These organisms can be found almost everywhere, and they're in very large amounts in those places. This is the bacteria you normally think of because it's the bacteria that makes us sick. But it can also be free-living and means that it doesn't even hurt us as humans. It can actually help us as humans. Now, the second prokaryotic domain is archaea. Now, archaea and these organisms in this group look a lot like the domain bacteria, but genetic studies, you looking at DNA, has shown that they're different. These organisms are known for living in very extreme environments. These are the extremophils. They like to live in really hot environments or really salty environments. Okay, so they might live inside volcanoes or anaerobic environments like swamps or even something like the Dead Sea. They live in areas where no other living thing can survive but them. The last domain is Eukarya, and it contains the other kingdoms that you've probably learned about: Protista, fungi, plantae, and Animalia. All of the cells that make up the organisms of domain Eukarya are eukaryotic, meaning they do have a nucleus and organelles. The first kingdom is Protista, and it's a very diverse group. Normally they're unicellular, but in this group they can be autotrophs or heterotrophs, meaning they can make their own food like through photosynthesis, or they can consume food like we do as animals. Some organisms of this group are algae, protozoa, and slime molds. Kingdom fungi is the next one, and this is mostly multicellular. They're heterotrophic via absorption. They're normally decomposers. Examples include mushrooms, yeast, puffballs, and of course molds. Kingdom plantae consists of multicellular plants, which means that they're going to do the process of photosynthesis. Examples would be like rice, roses, moss, ferns, pine trees, and that's just to name a few that are in the kingdom of plants. The last kingdom is Animalia. This is animals The last kingdom is Animalia. Animals are multicellular and they're heterotrophic, meaning they're going to consume or eat their food. Examples would be sponges, worms, an octopus, lizards, tigers, and again, these are just to name a few that are part of the animal kingdom. This semester, we will be looking at each of these groups in more detail as we move from chapter to chapter. Now that technology has advanced, we are able to investigate the evolutionary history of a group of organisms in more detail because we can actually look at their DNA. This is called phylogeny, and it looks for a common ancestor based on ancestral characteristics. Now, this is a characteristic that's shared by the new organism as well as the ancestor. Now, they're separated from the ancestor because they have what we call derived characteristics. These are differences that they've developed over time that are different from the ancestor. A cladogram is used to illustrate evolutionary history of a group. It's a picture. So if you look here, down here at the bottom of this particular diagram, you would see that the common ancestor is down here, and then a divergence happens where they have new derived characteristics. So you see the monkey over here has a tail and things like that, whereas your ape or your gorilla on the other side doesn't have a tail. There's characteristics that makes them different, but they were related at some point. So a cladogram is used to illustrate these. In your first lab, you're going to be asked to construct a cladogram based on certain characteristics in organisms. You're going to have a chart that's similar to this in the lab. You're going to need to investigate the traits, what they mean, and you need to also look at each organism and determine whether they have those traits or not. In this example, you will see that the notochord is the trait that all the organisms share, but the other traits are considered derived for different groups because they're not found in all groups. This is going to help you then construct the cladogram. So it is important to investigate the traits and the organisms first. Once you fill out the chart, then you will be able to construct the cladogram like you see here. You'll see on this cladogram that the characteristics are found on the lines, and on the end of the lines you'll find the actual organisms. Okay, so the whole idea of having a common ancestor, the vertebrae is the first new trait that tuna has versus that lancelets do not have. 4 limbs, you see it's first seen in the frog, but the tuna does not have it. So at each branch you see a new derived trait that leaves out organisms lower down on the cladogram. Now your cladogram in your lab is going to be a little simpler, and it's going to be more of a single line like you see here in this picture. Your cladogram will look very similar with traits on the line and then your branches coming up with your organisms. Now, evolution is change over time, and there are certain types of data that are used to trace out this phylogeny or evolutionary history. First is the fossil record. Fossil records have an advantage because they can be dated, and they can reveal relationships even with organisms that are extinct or gone. A disadvantage of fossils, however, is that the environmental conditions must be right for a fossil to be made. It has to be buried in order to make a fossil. Also, if the organism doesn't have hard parts like a skeleton or something like that, fossils are a little harder to form. Okay, and so you're going to have gaps or missing pieces in the fossil record because of this. Morphological data is another type of evidence where they look at the anatomy of organisms. Well, the first one they may look at is what we call homologous structures. Homologous means that they are similar in their anatomy or structure, but their function is different. Okay, this is an example of the 4 limbs of different mammals: a human, a cat, a whale, and the bat. And you'll notice they all have a humerus. They all have a radius and ulna. They all have carpals and metacarpals, and they all have phalanges or fingers. However, we use our hand differently than a whale uses its fin or a bat uses its wing. So they have similar structures, which makes us related to a point, but we use them for different functions. On the other hand, analogous structures have the same function, but the structures are different in their anatomy. Okay, so these are all wings, and they're all used for flight. But you'll notice how the structures are different. Over here in your insect, there's just a membrane. Okay, there's no hard parts to it. Whereas in your middle picture, that's a bird because there's modified feathers. Okay, so we see that they all overcame a certain obstacle for flight. They just did it in different ways. These do not necessarily show relationship or relatedness because the structures are different. Last is the molecular data, which looks at DNA and RNA. The more DNA sequence that two organisms share in common, the more closely related they are. If they have 60% the same, they're going to be more closely related than if one of them has only 40% the same as another organism. Now, this is a relatively new type of data because we just now have the technology that allows us to investigate this. But it's a great way of data to show evolutionary relatedness and maybe even common ancestors. This is what's also causing a change in the classification system. It's constantly evolving and changing as well as technology becomes better and better for us as scientists. Now, my goal in these lectures, guys, is to supplement your reading. Okay, to help you understand certain concepts. Okay, this is not to take place of your assigned reading. Okay, so keeping that in mind, you need to be reading your chapters in the book. In order to be successful and to succeed on your unit exams, you're going to need to read, and I hope that these lectures also help in that. However, if you have any questions or concerns, please let me know, and I will be doing these lectures for each chapter throughout your course. So good luck in Biology 124, and again, if you need any help, please let me know.